All right, so good evening and welcome to Kona Science Cafe. This evening we have Dr. Rick Bennett, who's going to be discussing microbial safety in our nearshore waters, including some interesting techniques that are being used to measure such the microbes or infer them. Rick Bennett's a consulting environmental and medical microbiologist. He's been living in South Kona for something like 20 years. And prior to that time, he was with the University of California researching microbial contamination of Northern California waters. He's the past chair of County of Hawaii's Environmental Management Commission, and he's presently the Kona Coast Waterkeeper, an affiliate of Waterkeepers Alliance International. All right, so please take it away.
Thank you, Rod, and welcome everybody. We're going to hit the tops of a lot of topics in the next 30 minutes. And many of these topics we could spend a whole hour on, but I kind of wanted to paint the bigger picture. And so we'll start out a little tongue-in-cheek because I like to have fun when we work. And I'm going to say the measurement of microbial safety of our recreational waters is a swag. And I think most people know what a swag is, but it has something to do. Why isn't we advancing the slides here? Here we go. Has something to do with one of these, otherwise known as a donkey, and is wild. And here's his guess. He's suggesting that we measure a bacterium called Methanobacter brevibacterium smithii, and can't even say it, nonetheless understand it. But this particular microbe in our gut is the one that makes methane gas. And it is unique to humans. And so actually people did propose that we measure this organism because it's not found in other animals. It doesn't grow in the environment. And so it could be a nice indicator, but it's fallen out of disfavor. So it's definitely a scientific wild ass guess. And so even though we're guessing, we are going to be okay. And I'm going to share with you some information about how the microbial ecosystem works in our environment that should give us some measure of reassurance that going into our waters, even though a lot of stuff we generate flows into it, we are going to be okay. So as you know, we have a whole lot of cesspits on this island, 50,000 plus. And on many locations, a cesspit is just a hole in the ground covered with concrete as a modern cesspit. In the old days, they were covered with boards. And in the very old days, they didn't cover them at all. These entities communicate with the groundwater and the nearshore waters. You'll notice the picture of the rhodamine dye at Puako. This showed up in the matter of hours. Similar observations have been made in the Florida Keys. And in fact, we're going to learn a lot from the Florida Keys because of the similarity. So all of this cesspit material represents about 30 million gallons per day of black water that is discharged to the ground where it magically disappears in most cases. The attenuation of microbes in this environment is not well understood, but given some new technology that I'll mention later on, we're getting a handle on it. But even more problematic from the perspective of microbial safety is the fact that we're overnourishing the ocean. The humans in the Greater Kona watershed add something around 400,000 pounds of nitrogen to the groundwater and to the ocean per year. About half of that amount is phosphorus. And the sad truth is that septic systems that are being proposed as the solution to cesspits do no better because they are not designed to remove nutrients. But there is hope. Again, the East Coast is leading the way. So here's a map that I downloaded from the County of Hawaii's GIS. And the green and the brown lines show you where sewer service is available in the greater Kailua-Kona area. And if you look at the insert that I blew over there on the right, let's see, look at the mouse here, you'll see there's a tremendous number of homes here and even right here and here that are not sewered. And we would like them to be. The problem is today, to dig a trench, to hammer a trench in the Blue Rock and in the Pahoehoe, according to the Department of Environmental Management Director, we're looking at around $3 to $4 million per mile just to dig the trench. And so connecting all of these homes to sewer as a retrofit, it seems rather unlikely unless a big source of money falls out of the sky. We also know that our sewer infrastructure is falling apart. For those of you that dabble in this sewer engineering, like I don't, but I do read some, all sewers leak, and it's as a function of their age. We have some sewer pipes now in the vicinity of on Kailua-Kona Bay that are 50 years old, and the engineers, the contractor has reported to the DEM that some of these iron pipes are completely rotten on the bottom. There is no pipe. And this may explain some of the problems we're seeing in that bay and along Ali'i Drive, where pipes are very old, perhaps fractured. The good news is, and you'll see my little good news icon all through the talk, is that the process to repair these leaky pipes is underway. It's going to be epic. There are ways of fixing these pipes without having to dig up the entire street. Time does not may go into that, but we can track where we need to work by measuring the chlorides in the wastewater. The chlorides in drinking water, 100 to 400, we're seeing levels of 600 to 2,000 milligrams per liter. This is an indicator that there's a leak, but it also means that this wastewater has got so much chloride in it that it's not feasible or impractical and perhaps even detrimental to use this reclaimed water for agricultural purposes. The chlorides are simply toxic. So here's one of the best diagrams of our water system. And this was done with data from our coastline, Honokahau Harbor Wells, Koloko, Honokahau National Historic Park. And under the coastal plain is a brackish water, which is a mixture of tidal seawater and fresh water that's moving down from the Malka conditions. And this water mixes and is heavily tidally influenced. When the tides are high, that change in height can be measured in these near coastal monitoring wells. Unlike the Hilo side, we don't have rivers and streams that we can see, but they're there. The dark blue in this slide is an infrared image taken by Craig Glenn and his team at UH Manoa. And the darker the blue, the colder the water. Notice how cold Honokahau Harbor is. Right here at the head wall, using isotopic markers, radon, natural radon markers, hydrologists have estimated that into that harbor flows about 3 to 3.5 million gallons of brackish water every day. And similar flow, big flows are occurring. If I could show you a map of the entire Kona Coast, there'd be 42 major submarine groundwater discharge sites. And what you can't see in this photograph are the little tiny ones that exist. Dr. Glynn estimates that for every mile of the Kona Coast, between 1 and 3 million gallons of brackish water flow into the sea every day. And this is how what we do on land communicates to the sea. So, with all of this water, stormwater runoff, wastewater percolating through the ground from intentional discharges or incidental discharges in septic systems and cesspits, it moves its way to the sea. And are the tools that we use today to assess recreational water safety, and I need to be a little careful here and say recreational water risk, because there will never be zero risk, but there are situations where the risk is high. It is difficult, however, to measure. The tools we use are antiquated, they're misinterpreted, the monitoring program is poorly funded and understaffed, and the risk is misrepresented. And hopefully by the time we're done, I'll be able to, you'll see why. And so the question that I hear all the time, and the question that I ask myself when I put my grandchildren in the ocean here a couple of years ago is it safe? And categorically, I don't know. I don't think anybody knows. But again, I think we're going to be okay, because we have been okay. We just have to be mindful. And so the first test that we're going to talk about is the Enterococci water test. It is a test of a genus of organisms that the EPA established long ago. And Hawaii gets about $1 million a year from the feds to implement the monitoring and posting provisions of the U.S. Beaches Act of 2000. It's a little history then on the fecal indicator bacteria. The acronym is FIB. It's very, very fitting. Back in the 1980s, we measured coliforms, thinking that they were mostly from the intestines of animals. We know they grow in the environment. If you want to grow coliforms, build yourself a compost pile. Then they came up with fecal coliforms in 1966, which is just a coliform test run at higher temperatures, warmer incubator that is. And that pretty much fell by the way because a lot of these organisms aren't fecal at all. And it's gone by the wayside. You'll still see fecal coliform tests in some of the literature and some of the regulations, but countries like Australia have completely thrown it out. So in about 1970, the EPA, because it has to calculate risk with its regulations, came up with the Enterococci for testing marine waters. For those of you who've been around for a while, you might know it as fecal strep. The problem is their risk assessment was done in waters with known wastewater discharges. And the other problem with the test is it will measure 50 different species of Enterococci, only 2 of which are common to the bowels of animals. And they themselves are not major pathogens. They're pathogens for people who have immune compromise. They're hard to count, and as pointed out by UH Manoa probably 20 years ago, they grow in the environment. They found it growing in this Manoa stream well above the town. And so it just doesn't work. So very quickly, how we count it today, there's an IDEX inter-alert system. You pour your water sample into the reservoir of this tray, you seal the tray, incubate the tray in an incubator, and then when it's out of the incubator, you put it under ultraviolet light and you count the colors. And then you take that count and look up the count on the most probable number table. And so let's say we have 11.1. It'll be 110 because the seawater is diluted 1 to 10 with fresh water. But the number statistically falls within the range of 6.1 to 20.5. In other words, 61 to 205, with the typical mean of 1.1, or in this case, 110. So that's the inner alert test. Now, when we apply the inner alert test then to determining risk, the way the EPA does that is measures illness in a bunch of swimmers in waters known to have a wastewater outfall. And they measured every day, and then they calculate the geometric mean, in this case 35, and associated with a geometric mean of 35, we may have 36 intestinal infections per 1,000 people recreating. And this number holds fairly well for water bodies like this one here, where they have a known wastewater discharge. It does not hold for coastal waters like ours, where there is no known major wastewater discharge in an area where you can do the epidemiological investigations. And so the bottom line is, we just don't know. And just to illustrate difficulty here, this is the geometric mean. And this is the statistical threshold value of the geometric mean. And this is a multiple observations over a 30-day interval. I am not aware when 30 days of monitoring has ever occurred on this island. They do individual monitoring, and if it's above 104 or so, they may post the beach or issue a warning, but seldom do they ever come back and do 30 days of sampling to establish what the geometric mean is. And so the risk of 36 per thousand in our situation just doesn't apply. And when it does apply, the statistical fit is not very good. Here's some real world data looking at enterococci counts and the relative risk of getting a gastrointestinal infection. Here's the scatter plots, and here's the regression line for those scatter plots. It has an R value of only 0.37 and just barely statistically significant. So this is a pretty sloppy estimator, when in fact we know that the real risk to people is not the bacteria, but the virus that are in the water. And the virus can be present when the bacteria are not, and conversely. So here's a little more of the confusion about what we're measuring. The arrows that I drew here represent measuring the two Enterococci that are associated with humans and animals. These others are in the environment. They will grow in the environment, and they will grow quite well. And so when we do the Enterococci test, we're measuring the whole gambit. And whether it's just beach water, or it's urban runoff, or sewer water, wastewater treatment plant influent, or wastewater treatment plant effluent, we're still dealing with a large proportion of Enterococci that are not associated with the human gut. Unfortunately, our Department of Health today on its website has this quote: untreated wastewater contains pathogens such as bacteria, protozoa and virus that can cause gastroenteritis, hepatitis A, conjunctivitis, leptospirosis, salmonellosis, and cholera. Unfortunately, that's just not true. Historically, sick people will shed these, and when they defecated in open toilets that ran in gutters in streets in Europe, outbreaks of cholera were common. But there hasn't been a case of cholera in Hawaii for over 100 years. And I haven't heard about a case of salmonellosis. Leptospirosis occurs. And that's, our wild pigs carry that, but it's not from swimming. And so there is just a great concern on the part of the health department, is we just have to make sure that this doesn't happen and make us look bad. So there's a huge bureaucratic risk aversion and an inattention to scientific data. And then most importantly, there's this, the yuck factor. It's just feces is yuck. It's just a disease waiting to happen. And the truth is, it's not. So now we have the ability to measure the human microbiome wherever it is. In this case, we're talking about the gut. And the Human Microbiome Project Consortium is a group of about 100 plus scientists from all over the world that developed the DNA technology to go into the gut, take a fecal sample, and characterize all the genera that are there and to characterize their density. And so what this article that was published in Science in 2012 said is that we looked at feces. We didn't find any evidence of pathogens in healthy people. We didn't find any evidence of pathogens in healthy people. Now the rules change dramatically when somebody is sick with, let's say, the norovirus, lovingly known as the cruise ship virus. They shed trillions and trillions of virus for a few weeks. But in a healthy population, the pathogens just aren't there. And so we've gone from an era of culture and grow microbiology, and I cut my teeth on doing just this. And many times, nothing would grow. And so what does it mean when nothing grows? Does it mean that fluid, that tissue, that blood was sterile? No, that's not what it means. What does it mean when nothing grows? Means nothing grew, period. No other interpretation can be made. Now, today, using the next generation sequencing and using the technologies like PCR that's used to detect just a few viral DNAs or RNAs in your nose, we now can bring that technology to ask the question, what is the microbiome of the human gut? What is the microbiome of wastewater? What is the microbiome of sewer water? And they're all different. They're different ecosystems. And so from a microbiologist's point of view, that's really good news. We are now able to differentiate the microbiome of animals from one another. And we can differentiate the sewage microbiome from the human microbiome. And looking for human pathogens as an indicator of sewage contamination of drinking water or recreational water, this work shows us that we've been looking in all the wrong places. This paper by McClellan showed that there are 3 to 4 major genera simply taper once wastewater has hit the sewer pipe. once wastewater has spent time in a septic tank. It shouldn't surprise us that that ecosystem is very, very different than inside our gut. And so McClellan is suggesting that we need to look for these sewage types as indicator species for wastewater leakage into drinking water systems or recreation waters. And I'm told that Dr. Tracy Wegner is going to be doing some work over on the Gila side, and some of the samples are going to be sent to Dr. McClellan for their next-gen sequencing to determine if that's kind of a sewer microbiome. So this is exciting, that we may be able to microbiologically finally detect the presence of sewage in drinking water, in recreational water, microbiologically. Well, I used to surf a long time ago, and I don't ever recall getting sick, but surfers are the canary in the coal mine. But it's not huge. You know, they ingest a lot of water surfing, but only a slight increase in gastrointestinal illness. Now they're out in deeper water, farther from shore, perhaps it's more mixed, who knows. But what's interesting is that skin infections are far more prevalent as a result of recreational water exposure than gastrointestinal illness. But skin infections have never been measured as part of the epidemiological studies. Here, this one Florida study said that if you're in the ocean, you're about 6 times more likely to get a skin illness. This is not a skin injury, a skin illness, than people who don't go in the water at all. And then there's the sand. People have got infected just sitting in beach sand. So here's a study done at UH where they looked at the decay of bacteria in the water column, which is here. Decays fairly quickly, but once you put it in sand, the death rate's relatively slow. Now there's no light exposure here, so, and this is a laboratory aquarium situation, but there's something about sand that gives microbes opportunities to set up shop. Here's some work done in Southern California beaches by a researcher named Goodwin, where they looked at several types of bacteria, including Staph aureus and Enterococci in beach water versus beach sand. And there is, for Staph aureus, there is a slight increase. But for Enterococci, look at the scale change here. This is the natural log. So these two scales aren't the same. So there's a lot more Enterococci in the beach sand. And we now know that when tide and surf events stir up the beach sand, and it just happens to be state test day, we can get an elevated count. And that count has nothing to do with a wastewater discharge. But we got to pay attention to staph. Hawaii has a lot of staph infections, and we don't know why. This man here, Dr. Alan Tice, has been studying clinical infections of Staph aureus in Hawaii for 20 years that I know of. And it's in the ocean, can be found, and it can be found in beach sand. And it doesn't correlate with Enterococci. And yet, children like this playing don't appear to be sustaining overwhelming numbers of infections, but they do occur. And the reason being is that there are many, many, many different strains of Staphylococcus aureus. 33% of us carry Staphylococcus aureus in our nose or in our perineum. And most of these strains have very, very low pathogenicity. And then there's something we call methicillin-resistant Staph aureus. This particular antibiotic-resistant staph used to be confined to hospitals, but it escaped. And Tracy Wigner in Hilo has shown a low prevalence of MRSA in beach sand in Hilo. Here's a photograph I took a number of years ago of a surfer, and we were having an information day along the coast. And the surfer came up to me and pulled off his bandage and goes, "You think I should see a doctor?" And I said, "The answer to your question is yes, and today." This is a pretty good size staph pustule, had it for a long time, and can be very threatening if it gains access to the blood. And so if a lesion like this occurs on anybody, you know, it needs to be seen, because it can be serious. The clinicians I have spoken to over the years say they're seeing these kinds of skin infections in Hawaii, in Kona, virtually daily. And they automatically treat them as if they're MRSA with a antibiotic antimicrobial cocktail that's usually pretty good. And they just treat them assuming it's the worst. And so far, so good. When this microbe gets antibiotic resistant to trimethoprim sulfa, we're in trouble. Now, in Hawaii, staph infections of the skin are not reportable diseases. That is to say, if a physician sees it, he's not required to report it as he's required to report a suspect TB case or a suspect case of HIV. So here's where the nutrients and the bacteria come together, and I see my time is getting ahead of me here. But this was a photograph taken on May 28th of the greening of Kailua Bay. And algal blooms occur all over the world for a variety of reasons. But one of the things that needs to be present is nutrients. And these nutrients may well be coming from a land source. Here is a marine upwelling that occurs almost all the time that causes this sand to be stirred up. And if this water is contaminated with the sewer water from the broken pipes that are in the immediate vicinity, nutrients are going to flow. And when the water is warm enough, you get algal blooms. And this is perhaps one of the worst algal blooms we've seen. So turbidity is sunscreen for bacteria. The ultraviolet rays of the sun do a magnificent job of killing bacteria in general. The sun helps control the bacterial biomass in the ocean. But in the nearshore waters, ultraviolet light is killing those organisms that may be of concern to us. And way back in 2007, I was asked to come down to Keauhou Bay and take a look at the water, and here's my paddle, and I can't read the name on the paddle for the turbidity that's occurring in that bay. And turbidity occurs in the bay for a variety of reasons, including surf events and surface water runoff carrying very, very fine sediments. But this particular event was a greening event that after a few days turned brown. And we know that as the transmission of light decreases from increasing turbidity, that the ultraviolet dosage to the water column, even down several feet, diminishes fairly quickly. So here's some good news. We're going to start measuring ultraviolet light at the beach. This is a temp sense device. It costs around $300, and it measures all kinds of weather parameters, including ultraviolet light. And the research has shown that when we accumulate about 20 megajoules per square meter, 20 megajoules per meter, that you get pretty dramatic reduction in bacterial numbers. Here's 10 to the 5th dropping down to under 10 to the 2 in one day. And so these days, I say that if you get 2 clear, sunny days, it's going to knock the microbial contamination that may have occurred after a brown water runoff event, return that water to what I would call low risk. This picture of Neil Mackay, who's sampling the water for bacteria, and notice how he takes water from the surface. And so if that surface water is exposed to this kind of ultraviolet, you're likely to get a negative result. And sometimes the bacteria are there, they just won't grow. They're considered viable but not culturable, B, B, and C. And once they're resuscitated with an environment that gives them the nutrients they need, they start growing again. And so ultraviolet light can be associated with false negatives. So here's what we're hoping to show, is that you have a rainfall event associated with risk. The County of San Diego today will publish risk warnings to the residents, to the surfers in San Diego County, just based upon a 1/4 inch of rain in a couple hours. Because they've established over the years that runoff events are associated with elevated Enterococci in the water. And so using this same technology with weather stations located at specific beaches, we'll be able to monitor the accumulation of solar energy. And these devices are net interactive, and we can write algorithms to warn us about a rain event, and another algorithm to tell us that we've accumulated 20 megajoules over a period of a day or two, and that will tell us that the risk has been reduced. This is probably going to be our best indicator of relative risk of recreation water safety on an ongoing basis. This is real time. You don't have to wait for 24 to 48 hours for some microbe to grow and be counted. And a lot of municipalities, including Auckland, New Zealand, are using this rainfall event risk model to warn the public that there may be increased risks associated with rainwater runoff. So some of the exciting things we're doing here, and I'll try to wrap this up quickly, is to measure sucralose, the artificial sweetener Splenda. It is a trichlorinated sucrose, 600 times more sweet than sucrose. It's in over 400 products, and it is extremely stable, like a lot of chlorinated organics. It does not degrade in the wastewater treatment plant. And like in ours, where it's exposed to intense ultraviolet light for weeks at a time. UV stable, no known microbial breakdown. That is to say, the enzymes that might break down sucrose just won't fit with those 3 chlorine molecules, and so you get no microbial enzymatic breakdown. There appears to be no natural analog. When researchers have gone upstream of human activities, they find nothing that shows up in the sucralose assay. It is not metabolized by humans. It is excreted mostly in the feces within 24 hours after ingesting it. And we can measure down to the nanogram concentrations using some technology called Orbitrap mass spectroscopy. The minimum detection limit is 12 nanograms per liter. It's an incredibly small amount, but in our waters, this is giving us some information. So here's some data from a preliminary study we did last fall. We got a sample of wastewater from the terminal pond, so the wastewater has flowed all through the treatment plant, so it's been out in the sun for days, and in that water we found 42,000 nanograms. This water is disposed of in a sump where it rapidly goes underground, and then the best science we have says that a lot of this water, if not all of it, flows to this head wall at the harbor where I indicated they've measured over 3 million gallons of fresh water, brackish water flowing into the harbor every day. Here we measured 48 nanograms per liter. That was our highest recording. At Kua Bay, it was below the detection limit. At the National Historical Park, it was below the detection limit. Kailua Bay was below the detection limit. Keauhou Bay was below the detection limit. Honaunau Bay was just at the detection limit, as was Banyans. There are very few homes in Honaunau, but they're all on cesspits, some of them very close to the ocean. The Banyans area along Ali'i Drive is on sewer. And so sewers leak, or there's somebody else has a discharge that we don't know about. They didn't connect to the sewer. They're still using their cesspit. We actually don't know if everybody along Ali'i Drive is connected. That information is not known. Down in Kahaluu Bay, this is an area where there is no sewer. We found 2 locations, one up by the church here and one here down by the pavilion, that had almost 3 times the detection limit, which according to the Florida people who've been doing this for some time, they consider that to be, unmistakably evidence of wastewater contamination. The other thing we're doing is looking at optical brighteners. Optical brighteners are put into laundry detergent here, and when the sun hits the brightener, a brighter color, a brighter white, a brighter blue is reflected into our eye. And so the laundry appears brighter and cleaner. When you take laundry detergent and put it up to a black light, it glows just like this. And so what we're able to do is to take ocean samples and stream samples, and using this spectrophotometer, which is designed to measure optical brighteners, we can detect them. Now, optical brighteners are decayed by ultraviolet light, and they decay very quickly. So the optical brighteners become a recent indicator, sucralose chronic indicator. The optical brighteners detects recent discharges because after in our sun, the ultraviolet decays. We can confirm that these are optical brighteners and not some form of dissolved organic carbon, which will also fluoresce, by exposing the sample to ultraviolet light in 2 iterations. And based upon this algorithm, we can say, yes, there's optical brighteners. So here's some very recent data. It's less than a month old. We identified OBs at the surfer access to Kahaluu Bay. My colleague, Dennis Mihalka, goes out on his kayak and takes samples. We've identified optical brighteners offshore now on 2 occasions, several weeks apart. And we've identified optical brighteners in Keauhou Bay at 2 locations. We have several locations we've tested that came up negative. Just this week, we tested along Ali'i Drive all the way up to the bay, Kailua Bay. Most of those samples were negative, as I would expect. Water was clear, sun was bright, but we did find a few, which is very interesting, and we're going to be repeating that. So I've gone over, I'm sorry. I just want to leave you with a thought. Just like medical people must always assume that the patients they deal with may be infected with COVID, they may be infected with TB, they may be infected with hepatitis B or HIV. You have to assume. And so they adopt what they call universal precautions. This day and age, I'm proposing that we need to bring that concept of universal precautions to our judgments about whether or not we're going to use the ocean in various places. First and foremost, don't recreate in stormwater. You hear about brown water advisories all the time in Oahu. We had brown water advisories when I was a teenager and surfing along LA beaches. And sometimes we surfed anyhow, and the stories of illness are significant, but they're just stories. If you see a floating greasy scum, it could be natural, or it could be from a cesspit. I swam into a cesspit scum in Honaunau Bay about 10 years ago. Open wounds in the water are a prescription for a staph infection, and I speak from experience. I almost lost a finger. Ocean water is not healthy for tissue. It's far more salty than physiological saline, and it kills tissue. And that dead tissue is food for staph. Vigorously clean and medicate all wounds created at the sea. And I do mean vigorous. Turn the faucet on full blast and just blast that wound. Irrigate it. That's what they do in the ER. And then watch for signs of infection. And if it gets infected and it starts to fill with, secretions that are pus-like, and it gets larger, do seek medical attention. Wash off seawater and sand, especially sand. I watch these kids play in the sand. I think, okay, mom and dad, do a good job of washing that sand off. And then gastrointestinal disease lasting more for a day or two, especially those with fever, seek medical attention. And critical is a naturally occurring Vibrio that attacks people who have compromised immune systems. It will cause widespread bruising and fever and swelling of the entire limb. This is life-threatening, needs immediate medical attention. We have a lot more research to do, and we're going to do it. And if you want to join in and help us with sweat equity or financial equity, we're all ears, and our hearts and minds are wide open to all the help we can get. And as I put my grandchildren in Kahului Bay several years ago, I was thinking about this, and I determined that the risk was minimal. But as we go down the track and more of us live near the coast, the risk is going to increase. And I think we know we need to know. The question is, will we make these young people proud of us for doing the right thing? Thank you for listening.
Excellent. Thank you very much. So are there any questions? Okay, chat. Where'd my chat window go? There we go. I don't know. I guess just go ahead and unmute if you have a question, because we don't have lots of people stepping over each other.
Don't be bashful.
Yeah.
I don't bite.
Hello?
I'll go ahead and break the ice.
Okay.
This is Dee Fulton, a colleague of Dr. Bennett's, I'm happy to say. And Dr. Bennett, we had this dramatic multiple episodes of algae growing in one of our favorite swimming spots, Kailua Bay. And some of the things that I picked up from your talk tonight, just to reinforce, is that turbidity and algae can act like an umbrella to bacteria and viruses. So I guess if we're seeing algae in the bay, we probably shouldn't be mucking around in there, because that's putting a roof over those potential pathogens.
Well, your synopsis is correct. What we don't know is the density of the pathogens. I think the available research is saying that pathogens in our ocean is extremely rare. Now, if there's an outbreak in a household of somebody's got the cruise ship virus, and several people have got this profuse watery diarrhea where people shed hundreds of trillions of virus daily. Now, if that's getting into the ocean and you swim through that and you get a couple of viruses on your lip, that's an infective dose. 1 to 10 of these viruses is an infective dose. But it's just not exceedingly common. We hear people getting skin infections, we hear people getting eye infections, we hear people getting gastrointestinal infections. And what we don't know is if it truly came from the ocean, because we're not able to measure the pathogens in question. Now, if our indicators that truly represent sewage are saying that there's a problem with sewage contamination of the recreation water, then we need to take that into stride and say, okay, today is not a good day for swimming. But just because the water is exceptionally cloudy, does that constitute an unacceptable risk? I'm asked this question all the time. I don't know. But it certainly increases the risk. Is it hugely significant? I don't know. I don't think anybody knows. I'm not even sure that's knowable.
Okay. Let's see. Doug had a question.
Yes, Dr. Bennett, concerning the nitrogen and phosphorus pollution that's so damaging to the reef, the state is mandating a conversion to septic systems, which does nothing to address that. You mentioned that the East Coast had some solutions to that. What's in the works? What has anybody found as far as ways that we can treat our sewage that will remove the some of the nitrogen and phosphorus?
Thank you for your question. This is another presentation. Starting in Long Island and working up to Massachusetts, up into Maine, and now down in Florida, the engineers and the public health people have discovered that if you bed your leach field, which takes the water out of your septic tank, and you bed that leach field in 18 inches of sand and sawdust, and on top of that, just another 18 inches of sand, and then you put your distribution pipes on top of that, this microbial system that you're building will remove easily 70% of the nitrogen and phosphorus, and in some Florida studies, as much as 90%. I'm in conversation with an engineer who's been working on this since the late 1980s in Florida, and he has written statutes for the Department of Health wherein they specify how to build these nitrogen and phosphorus removing leach fields. As we speak, we have an entrepreneur in South Kona, and he found an engineer here, and we are designing what's called the layer cake system for a home on Kealakekua Bay, and it's right on the bay. And we will put devices in that system to measure the nitrogen and phosphorus coming out of the septic tank, and then the nitrogen and phosphorus that is leached through the layer cake. And again, with our warm temperatures, we expect good performance, because as they've learned in the Northeast, when it gets really cold, these systems don't work too well. Microbes don't like cold either. But in Hawaii, like Florida, the expectation is they're going to work quite well. And it's going to add a very small increment of cost to putting in a conventional septic system. And this will be a septic system that actually works.
Okay, I think Bruce had a question if you want to voice that.
Can you hear me?
You can.
Yes, yes.
Oh, great. Well, I wrote it down. It seems to me like you established that human sewage really, or human excrement really doesn't present a problem from healthy people. We know there's a large yuck factor there. But
Yeah.
then you jump to sucralose as an indication of such sewage, implying that that is a problem. Is there really a problem with green water? I swim in Kailua Bay all the time and have decided, unless it's so green I can't see, to do it, and I don't seem to get sick.
Sucralose is simply going to be an indicator of where sewage water is being released into the environment when it shouldn't be.
Right.
To release sewage water into the environment requires a permit that you'll never get under the Clean Water Act.
Sure, I understand, and there are leaks and stuff, but does that really present a problem beyond the yuck factor?
Perhaps not, but it could be a significant source of nitrogen and phosphorus, which alters the nearshore marine ecosystems.
So then normal ocean, the balance of normal ocean bacteria will be altered by excess nutrients of one type or another.
There's no question that the bacteria may respond, but what clearly does respond is the phytoplankton. Photosynthetic microscopic algae.
Yeah. And that's really what causes the green, correct?
Indeed. Now, it's interesting. I was channel surfing this weekend and saw a program called Voice of the Sea. And this spring and summer, Southern California had huge problems with red tide. The ocean turns red-brown and it stinks. And it's attractive because it glows at night when the waves break. There's a phosphorescence. But they also believe that nutrients and much warmer ocean temperatures than normally occur in Southern California were factors.
But again, we don't know beyond the yuck factor if it's really a problem.
No, we don't. Now, given the PCR technology and given the next generation sequencing technology, we could go into the ocean where we believe we have no wastewater leakage and confirm it. And similarly, where we believe we have no leakage, we can confirm that we do or do not. This work was done on the Russian River in California, that is a huge recreation river in the summertime. And they found that the Enterococci didn't work in that river at all. And that is to say, when the Enterococci were very low, there were large numbers of human fecal organisms in the water. And what they didn't measure, and what's very difficult to measure, is the actual pathogens themselves. That requires a very specific PCR. And Manoa is doing some experimental work on that, and they're getting encouraging results. The difficulty with PCR is it's very expensive. It requires special laboratory facilities, special operators, and to be useful, as we're learning with COVID-19, to be useful, that data has got to be available within 24 to 36 hours. 48 hours, 72 hours, it's so after the fact that the data is meaningless.
Right.
So that's why we think that looking at runoff events and looking at intense UV events will be our best estimate of when risk is elevated and when it's been depressed.
So Pam had a question.
Thank you.
Along the lines of sucralose. Okay, so the question was, it scrolled up on my window here. the sucralose sample that was in Kailua Bay was below the detection limit, right?
Correct.
So essentially zero. Do you have an explanation for that? Because it seems like there are broken pipes in Kailua Bay.
Had we done detailed transects in the bay, where you take near shore out 10 meters, out 50 meters, and actually kind of develop a sucralose map of the bay, we might be able to explain why we didn't find it. The samples were taken fairly close to the shore where people use the bay. And at that particular time, it was below the detection limit. And so to get above the detection limit means there's got to be, a reasonable supply of sucralose moving into the water. Now we know that the bays do not uniformly mix. There's stratification, all types of stratification going on in the bay. And so it may be that where that sample was taken was in a protected eddy that didn't receive the groundwater discharge. I speculate aloud. It's hard to explain negatives. It's all we can say is we didn't detect it. When we do detect it, however, we can say that that is evidence of human waste activity, unless there's somebody pouring sucralose into the ocean for fun. And that could give us a false positive.
Okay, Andre, you still here? Not sure he's still here. Anyway, great presentation. I noticed surfers wouldn't enter the water 2 weeks ago at Banyans because so many surfers exclusive to that location were getting sick. This is the first time I've ever witnessed this. And thank you for the presentation. And then Kathy, who I think is still here, had a question.
Yeah, well, actually it's for me. I'm John, her husband. The questions were, I figured out another one. Where are the locations and the frequency of these tests that are done in the bays?
By us or by the Department of Health?
The Department of Health.
They have a standard run they do. And I think they start down in Honau now, and then Keaho, Kahaluu, Banyans. And they may jump up then to Kailua Bay. They go up to Makalavena, Kua Bay, Manalani Beach, Spencer's, Hapuna, and sometimes even Mahu Kona up in the far north.
Right.
And they're supposed to hit them once a month.
Once a month?
Once a month.
That's not very funny, actually. But so
even if they hit them once a week, they still could miss an event.
Right. Yeah. That's what I'm seeing here with the runoff. I have a second question that came up. I worked for the drinking water program in the state of Oregon for my career, and when I retired a few years ago, they were looking for surface water systems that had treatment plants, caffeine and pharmaceuticals as indicators for drinking water quality in surface rivers. Is there any?
A lot of work has been done with caffeine, conitine, which is a metabolite of nicotine, pharmaceuticals, antimicrobials, antispasmodics. The difficulty with a lot of these is they're fairly labile, that after days, hours, weeks in the environment, they break down. And the exciting thing about sucralose is it's just so doggone stable, undigestible, not degraded. The Swedish have adopted sucralose as their sewage indicator.
Okay, all right, thanks.
That seems to me that eventually the noise floor is going to.
I've got a question.
Up, right? Go ahead,
I have-
Kelly.
I have a question.
Go for it.
Dr. Bennett, when we are using sucralose as an indicator from sewage spill or whatever, has any consideration been given to the fact that at Kahaluu Bay, probably every one of those people swimming out there is peeing in the water?
Yes, we have discussed that. And it turns out that about 85 to 95% of the sucralose is only excreted in the feces. And if you're absorbing sucralose out of your bowel, you probably have what's known as leaky bowel disease.
I just think that virtually everyone that swims out there does pee. I don't know about yourself, but I can speak for myself that it has happened. And I think that that could, if it's going to be that persistent in the environment, that it probably is there at least for several months. And there's quite a population of people that use it.
Let me say it again. Sucralose is by and large not excreted in urine.
Okay. Okay. That's my question. Thank you.
And if you go to the doctor and they detect sucralose in your urine, there's probably something going on with inflammatory bowel disease.
Thank you.
All righty. Someone a while back asked a question whether the presentation slides would be available.
The slides,
Mhm.
I can get you those slides in a PDF version.
That'd be great.
I'll have to put it on the cloud and you can download it because it'll be a file of about 60 plus megabytes.
Okay.
It's because of all the photos.
Yeah.
Yeah.
Okay. Great. Well, thank you very much. Any other questions? I guess not.
Well, if you'd like me to come back and expound on one of these topics that I kind of glossed over today, I'd be happy to do that.
Okay, great. Well, thank you everyone for showing up, and yeah, we'll do some silent muted clapping.
It's been my pleasure.
All right, have a good evening, everyone. I'm recording this, and I'll put it up on the website, and we'll figure out a way to make the presentation available to people who want it.
Hey, thanks so much, Rod, for launching Kona Science Cafe virtual. Really appreciate it.
Thank you.